Welcome, everyone, to our review for our last exam. I know it's classified as a final, but it's non-cumulative. It will cover reproduction and development and inheritance. So I've compiled a file for you guys. I know I just posted it a little bit ago, so if you haven't had the chance to scamper over to Canvas and download it, please do so. It's under the announcements section. And that's the file that we will be following along with. It says 152 152 final review, I believe. 152 Exam 5 Study Guide. All right, so we'll be talking about sexual reproduction first, and we're going to dive right into the male reproductive system, which is how it was divided. We will start primarily I'm just going to focus on the pictures because I feel like we've talked about a lot of this kind of end over end in these past couple of days. So this is the male reproductive system. One of the major differences is that the male reproductive system has a much longer urethra than the female reproductive system. It's going to be divided into 3 different sections, including here is the prostatic urethra, which runs through the prostate, and this is the intermediate urethra, and then this is the spongy urethra. In the female, we only have one urethra. It's very small, it's much shorter, and it does not contain anything other than urine. It's going to be separate for the reproductive pathways and the urinary pathways. In the male, the reproductive pathways and the urinary pathways are going to use the same exit. So the ejaculatory fluid and the urination is going to come out of the same pathway. All right, so let's see, what else are we looking at here? Here is the rectum and the anus, which would be just posterior to the reproductive system. We also are going to have the urinary bladder just to kind of orient you. Now, the vas deferens is going to connect the testes, which are going to be what creates the sperm. Remember, they're going to enter into the epididymis for transit. That's going to take about 2 weeks, where they're going to enter into maturation. They're going to learn to swim. So up here, we're going to have sperm that cannot swim. Here, we're going to have fully functional motile sperm. They're going to be able to be ejaculated through the vas deferens. Now, the vas deferens is going to run behind the ureters. Remember, those are going to connect up to the kidneys and run into the urinary bladder. So it's going to run just behind that. And then it's going to run here into the urethra. It's also going to be joined by other fluids, including fluids from the prostate and the seminal vesicles. All of that's going to mix together to become what we call ejaculate, right? Also coming out of this is going to be fluid from the urinary bladder. Let's see, what are we missing? I didn't really talk much about the penis itself. The penis itself is going to have multiple sections, including, here's the glans penis. And if we are uncircumcised, there would be the prepuce. Yes, I'm sorry, you have a question?
Professor, we can't see your screen.
Oh, I haven't shared it. Sorry. How
No, you're.
did
Thank
I
you.
lose you guys? All right, let me go back to Zoom. Here. Huh, I actually don't see you guys. I can hear you, though. How did I... Ah, here we go. Okay. Sorry about that. Thank you for bringing that up. All right, how about now? Now I'm screen sharing?
Yes.
All right, I apologize. Those of you who are
Thank you.
following along at home, though, you're able to see the whole time anyway because I'm just viewing my screen. So, all right, so now I'm back to, hopefully you guys are all caught up to where I am. I'm just on the first slide, or the first, I guess, page of the Word document. When we were talking about the reproductive pathway, I was pointing out the urethra and how it's combined into 3 sections in the male when it's only 1 in the female, that it's going to combine both the ejaculatory secretions and the urinary secretions down the same exit routes. We talked about the testes, which is where sperm are created, and the epididymis, where sperm mature about 2 weeks, and then they'll exit through the vas deferens, where they'll be joined by fluids from the seminal vesicles and the prostate, and then exit out through the spongy urethra. If we're talking about the penis itself, we have the glans penis, which is the tip, or what we would call the head, and then the body of the penis is split up into, it's actually only 2 different types of tissue, but there are 3 of them. So we have the corpus cavernosum penis and the corpus spongiosum penis. And these are going to be erectile tissue that will fill with blood to allow for the erectile event to occur. I'm going to show that here. So when I say there are 2 different types of tissue, but there are 3, that's because we have 2 of the corpus cavernosa penis on the top and only 1 of this corpus spongiosum penis. But they're both going to be highly vascularized, so we're going to be able to have blood flow in and out of this tissue to allow for the erectile event to occur. And then also right through here, this is the spongy urethra. It's actually going to run right on through the corpus spongiosum. So that's actually going to be the route that the fluid exits the body. So it's not directly in the center, it's actually towards the lower parts. We also have a pretty good view here of the testes. So this here at the bottom, the skin that covers the scrotum, is going to be the raphe. or the line, I mean, that goes right in between the skin that separates the scrotum is called the raphe. And then we have a muscle that's connected right underneath that. It's called the dartos muscle, which can allow for testicular ascent if it gets too cold, or descent if they get too warm, so they can moderate their temperature. Remember, sperm have to be created and maintained at a temperature that's a little bit lower than the internal body conditions, so they have to stay outside the body in order to be fully functional. We also have this cord here, the spermatic cord. Inside here, it's going to be right in that superficial inguinal ring. Inside here, we have another muscle called the cremaster muscle. And we also are going to have the vas deferens, which is going to run again from the epididymis all the way up, then across the urinary bladder, and then back down again out, matching with meeting with fluids from the prostate and the seminal vesicles, etc., for ejaculation. So that's going to be the route that the vas deferens is going to take throughout the body. It also is going to be highly vascularized in this cord. We have the testicular artery, and we have a vein. We also are going to have a lot of nervous tissue. So this is basically going to be by the way by which this external kind of organ is going to be connected to the internal blood supply and nervous tissue. Let's see. Okay, I'm going to move on unless we have any questions there. So let's take a look at the testes itself. So this is where sperm are going to be created. So sperm are going to be created in something called a seminiferous tubule. That's this little tubule here that's got all these awkward twists and turns. At the end of the seminiferous tubule, we have a region called the straight tubule. And then they're going to enter into the rete testes. From the rete testes, they're going to enter into the epididymis. We have three major sections of the epididymis. We have the head, the body, and the tail, or the corpus caput and cauda. Remember that sperm from the corpus and caput are not able to, they're able to fertilize an egg if they were put right next to it, but they're not able to swim up to it for the fertilization event to occur. Fully mature sperm are going to be found here in the caudal epididymis, where they're going to then be sent out through the vas deferens, which is found inside the spermatic cord, for ejaculation. And we did this in lab. We took testes and did a dissection. So if you were unable to join us for that lab, there's a nice video posted as well. Okay, I'm going to move on there unless we have any questions. The next thing we'll talk about is the creation of sperm. So sperm are going to be created, again, that happens in the testes, in the seminiferous tubule. So now we're looking at a section of a seminiferous tubule. And we're going to see cells that are going to be the spermatogenic cells. That means they're going to be turning into sperm by first becoming the primary, then the secondary spermatocytes, eventually a spermatid, and then a full-blown sperm cell, which is also known as a spermatozoon. And then that'll be secreted into the lumen of the seminiferous tubules. But we're also going to see some other cells here. Then these are called Leydig cells, or here it's substenticular cells. They're going to be cells that are going to nourish these spermatogenic cells. So they're going to be able to be there to help transform them and also to help feed them, because once we get to this point, sperm are not creating any of their own anything anymore. Their DNA, remember, here's our nuclei, it's getting more and more and more condensed until we're actually not using it for anything at all. It's just completely compacted and only is going to be unraveled once it finally gets to the fertilization event in the egg. That means that from this point forward, everything that the sperm uses is either going to have to come from the outside, or it's going to have to be fuel that it already has at this point. So we're going to be able to get things from, for example, the female tract called capacitation, or through epididymal transit. So the sperm are going to mature by getting things from the external tissue, but they themselves are not able to create any RNA or protein anymore. The other thing of note here in this diagram is that we start off with 2N and then we end up with N. And what does that mean? That means we start off with a diploid cell and we end up with a haploid cell. The haploid cell is only going to have half of the amount of DNA as the diploid cell, but a very precise half. It's not a random half. We have one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3, one copy of chromosome 4, etc. And so then it's like taking a deck of cards and making sure that each person gets 2 jacks and 2 queens and 2 kings and 2 aces, etc. And then when you put 2 decks back, or 2 half decks back together, you get a full deck, but it's going to be a little bit different than the original deck. And that's how we're able to take the sperm and the egg and combine DNA from our mom and our dad and create genetically unique individuals. But I digress. So this is talking about the process of spermatogenesis and how we start with a spermatogonium, which again is a diploid cell, and end up creating a sperm cell, which is a haploid cell, which will be ready for the process of fertilization after it undergoes a very long journey. So this is a little bit more in depth about the process of creating a sperm cell, or spermatogenesis. And remember, every time that we have what's called a stem cell, these are going to be basically stem cells or precursor cells. Every time that we start the differentiation of a stem cell, we're going to create another one that will end up being a reserve. So basically we'll have a division, and one of the products of division will remain a stem cell, and the next one will enter into the products of that's pathway of differentiation. So here's our primary spermatocyte. At this point, we're entering into a meiotic event. We enter into meiosis 1 and meiosis 2, which is going to take us down to the haploid state. This means that each set of these is going to have 1/2 of the DNA of the adults, and that genetic complement will be restored once it fertilizes the egg, which also has 1/2 of the adult DNA. Of note is that the sperm are going to share information across what's called a cytoplasmic bridge. That means that if this sperm cell has a different set of DNA that might be weaker than that set of DNA, it's okay because it's going to be shared. All of the information that's being created by both of these during this time frame is going to be shared until we get to the point where the sperm are actually going to be their own spermatozoa. And at this point, they're not going to have any sharing, but they also aren't going to be able to create anything anymore either. So they're all going to have a fair fighting chance. It's going to kind of level the playing fields. All right, so this is an image of what the sperm look like. The sperm are split up into 3 major pieces. The head, the mid piece, and the end piece of the tail. So the head is going to comprise the nucleus, and then on top of the nucleus we have this acrosomal cap. And this is a very important region of the sperm because it's going to have some enzymes that are going to be involved in breaking down hyaluronic acid. Now, hyaluronic acid is going to be what the cumulus cells of the eggs are held together by, so it's going to be one of the final barriers of fertilization that the sperm have to go through. And when they get to the point where they're entering that cumulus layer, this acrosome will rupture, causing what's called an acrosome reaction. All those enzymes will spill, and the sperm will be able to penetrate through and get to the zona pellucida of the egg. Again, the first one to the zona pellucida wins. After that, the zona pellucida shuts down, and we're not going to be able to have a second fertilization event occur. So the very first one there is going to be able to get this nucleus into the egg, and that's really the end goal, is the fertilization events. But the swimming, so this whole, all the head holds basically is the information and those acrosomal enzymes for penetration. But the swimming all comes from the tail. And in the tail, we're going to have these specialty regions or microtubules. They're very long, and they're basically going to shift left and right alternately, which is going to allow the tail to, well, flagellate, right? It's a flagella. And inside here, we have the mitochondria. Now, unfortunately, gentlemen, that means that the mitochondria don't ever make it into the egg during the fertilization event, which means that I digress. That means that the mitochondria is only ever going to come from the maternal lineage. That's not tested. I will progress here. Again, the tail is split into 2 major regions, the principal piece and the end piece of the tail. All right, so the testes are under hormonal control. They're going to be constantly creating sperm, and in order to do that, they're going to need to have information and context cues from the brain. So here we have the hypothalamus of the brain, which is going to gosh, that noise is annoying. So here we have the hypothalamus of the brain, which is going to be connected here to the anterior pituitary. Again, that's by that infundibulum region. And it's going to secrete, or at least the anterior pituitary is going to secrete gonadotropes, such as FSH and LH. Now, FSH is going to induce spermatogenesis in the spermatogenic cells, right? LH is going to stimulate testosterone secretion, and testosterone is also going to increase spermatogenesis. Testosterone can also be converted into dihydrotestosterone, which is something that was very important in the male before birth for the male patterns of development, and also is going to be very important for puberty and anabolism, which happens after puberty. But after we get to the point where we're creating testosterone at the puberty time point, dihydrotestosterone is not going to be as important anymore because testosterone is a much stronger stimulus. Anyway, these are both going to induce the spermatogenic cells to undergo spermatogenesis, right? So testosterone, FSH, all that is going to stimulate spermatogenesis. We also have inhibin, and inhibin is going to decrease the release of FSH. So that's going to have, and testosterone also is going to be an inhibitor. So the end products of these pathways are going to inhibit the upstream initiators. So testosterone, for example, regulates GnRH and the gonadotrope levels, and this is what helps us from getting too high or too low in our hormonal levels so that we can adequately create sperm at a continuous rate. All right, any questions on that? So testosterone is important for a lot of things, not just spermatogenesis, right? Testosterone is in the initiation of puberty and bone growth, and but also in the purpose of sperm production. It's involved in sperm production and sperm maturation. All right, so here's a negative feedback loop. It's going to talk about how we maintain our blood levels of testosterone, which is pretty much going to summarize the figure that I just showed you. basically what happens is when we have a stimulus, such as increased testosterone levels, um, testosterone is going to have this receptor in the GnRH cells that are, I'm sorry, the cells that are going to create GnRH in the hypothalamus. And remember, testosterone inhibits the creation of GnRH, so it's going to then decrease GnRH in our portal blood, which means that we're going to then end up with less LH in our systemic blood, which is going to affect the Leydig cells to secrete less testosterone. So that means that we're going to have a reduction in blood testosterone, which means that we're going to stop the inhibition of testosterone and turn it back on. And so in this way, if we get too high, the testosterone inhibits its own creation, and if we get too low, the lack of inhibition turns it back on. So this is how under normal conditions we can have regular levels of testosterone in an adult male, which of course helps regulate the control of spermatogenesis as well as other male reproductive and not just reproductive, other male characteristics such as protein anabolism, etc. All right, so the reproductive system that we just talked about is going to include the testes, which is broken up into seminiferous tubules, straight tubules, testes, and then the epididymis, which is broken up into corpus and cauda, or head, body, and tail. Then the vas deferens, which is going to be the pathway for ejaculation. And so this is going to kind of show you the pathway of the vas deferens. So here we have, so here's the posterior view, so we're looking from behind. So here we're going to have seminal vesicles secreted into the ejaculatory, into the seminal vesicle duct, into the ejaculatory duct. We also are going to have the vas deferens running into that ejaculatory duct. Remember, the vas deferens is going to run down the back of the urinary bladder. So it's going to run up along the entire spermatic cord, then back over the urinary bladder, loop back down, and then run behind the urinary bladder to meet the seminal vesicles and the fluid from the prostate to enter into that ejaculatory duct for ejaculation. All right, some other things of interest here are going to be the bulbourethral glands. These are known as Cooper's glands. These are going to help with secretions, also for seminal secretions, basically. Just really more lubrication than, so the seminal vesicles are going to secrete things that are going to be sugars and other, basically Gatorade for the swimmers for the marathon they're about to embark upon, whereas these glands are going to be more secretory glands for something like mucus to be able to help lubricate for the actual active copulation. What else did I miss? Again, here's the prostate. Then this is the prostatic urethra, which runs through the prostate and going to then interact with and become the intermediate urethra, which enters into the penis to become the spongy urethra. Yeah, okay, so I think I've touched on, oh, I did not tell you what these were, but you should know these are the ureters. They feed into the urinary bladder. So as I mentioned, that the vas deferens goes up over the urinary bladder and then feeds back down. It actually is going to feed right on through those ureters before it comes down to meet the seminal vesicles. All right, so we talked about the spermatic cord as well. The spermatic cord is going to be the structure that's going to hold things like the vas deferens, the arteries, the vasculature, the nervous system, etc. Lymphatic tissue is also going to be running in there, as well as the cremaster muscle. So we have two major muscles, the cremaster muscle and the dartos muscle, that are going to be involved in testicular descent and also ejaculatory, like they actually pulse during the ejaculatory events. For sterilization, men will have a vasectomy. Basically what that means is that they will cut the vas deferens. They'll go through and they'll do that in the testes generally, or at the top of the testes, and they'll pull it out. They'll make a small incision in the scrotum, and then they'll pull out the vas deferens and they'll cut it and ligate it on either side. Although ejaculation can still occur after this, there's no sperm entering into the ejaculate, so the ejaculate is going to consist solely of secretions from the seminal vesicles and the prostate. So fertilization cannot occur, but fluid will still come out. So the ejaculatory duct is going to be combined from many different the junction of many different ducts. Comes from the seminal vesicles, the vas deferens, etc. And basically the point is for us to be able to get the ejaculate, which includes the spermatozoa, into the prostatic urethra to join with fluids from the prostate to then enter into the intermediate urethra for release out of the spongy urethra. Again, the urethra has three different sections: the prostatic, membranous, and spongy urethra. We also have accessory glands such as the seminal vesicles. We talked about those and what they do. They basically are going to add, it's sugar, it's a fructose that's being produced so that they sperm are able to use that for ATP production. It's basically like handing out Gatorade to the swimmers. Additionally, there's some alkalinity of the fluid, and that's to help offset the acidity of the vaginal canal and the female reproductive tract. I also talked about the prostate gland and the bulbourethral gland, which are involved in secretion of acidic milky fluid for the prostate is going to have proteolytic enzymes that are going to help the sperm penetrate through, as well as PSA, which we use when we look at prostate cancer. So people will specifically at normal PSA levels versus prostate cancer levels, and they're going to be very different. And so looking at PSA can let people know what your stage of prostatic cancer is and give you an idea of your percentage chance of recovery when you are treated. There are also some hyaluronidase in there, which helps take the coagulated semen. So when semen first comes out, it's going to be kind of gelatinous, and then it's going to liquefy over the first hour and combine with all of these different secretions. And the hyaluronidase is going to help do that. The hyaluronidase is also going to help, which it's going to also be contained inside the acrosome. It's going to be involved in the release after the acrosome reaction, which will help penetrate through the cumulus cells of the egg. All right, so that brings us to semen, which is a mix of both the spermatozoa, right? So all of the fluid that comes out of the vas deferens is going to include spermatozoa, as well as the secretions that come from all the accessory sex glands. And again, that fluid's going to be kind of segmented. One's gelatinous and one's clear, and then they're going to end up liquefying inside the female tract to allow the sperm to be able to basically mix with the Gatorade and give them that extra boost that they need to enter onto that very long journey that they're about to embark through the female tract prior to fertilization. All right. Did I talk about this one? I did, right? I showed this one previously, so I guess this is just kind of the same image shown again to talk about the penis itself. The penis itself has three major sections. Again, they're two different types of tissue, the corpus cavernosa tissue and the corpus spongiosum tissue. Corpus spongiosum is where the spongy urethra is going to be, where the actual ejaculate is going to come out, and it's all vascular tissue, so all of this is going to be able to take in blood. And again, this is the second muscle that I was talking about, the dartos muscle here, which is actually going to be right underneath the testes and inside the scrotum. All right, so here is a frontal section of the penis going all the way back to the urinary bladder. So the urinary bladder is going to sit just behind the prostate. The urethra is going to feed out of the urinary bladder as well as also from the vas deferens feeding into the prostatic urethra. On either side of that, we're going to have the bulbourethral glands. Again, these are going to secrete mucus for lubrication. Then we're going to enter into the root of the penis. The root of the penis actually has the bulb of the penis at the very base of it. On the outside of it, we're going to have the crust of the penis. This is what's going to enter into the inner thigh, right? So this is the skin that's going to connect with the rest of the leg. Inside the body of the penis, again, we have the corpus cavernosa, which is going to be on the top section, the corpus spongiosum on the bottom section, and running through that, we have the spongy urethra. Again, the spongy urethra is going to be running through the corpus spongiosum penis. At the tip, we're going to have the glans penis, which has the corona at the very bottom here, you can see. And if we are uncircumcised, we may have this foreskin or the prepubis layer that will cover all the way back to about here. Of course, in first world nations, oftentimes this is removed in males for the purposes of generally preventing infection. This tends to be an area that tends to have things get accumulated in there and infection can occur. So oftentimes it's something that was removed specifically for the purposes of preventing different types of diseases. All right, so what did I miss? I think we touched on that. Okay, so here's going to be a transverse section of the penis. Again, I'm going to note that we have two different types of tissue, but there are basically three sections. Two of the sections of the corpus cavernosa penis, and one is the corpus spongiosum, through which the spongy urethra is going to run. Keep in mind this is highly vascularized, which means we're going to have the deep artery that's going to run through, and the spongy tissue here is going to basically just absorb that blood whenever the blood is turned on. Like there's basically a faucet, right, that's going to allow the blood to run into here. Once it runs into this deep artery, then it's going to seep out into all this spongy erectile tissue, causing the erection or the erectile event. When that blood is turned off, it will eventually seep back out for drainage, and then the erectile event will end. So this is the vasculature. If you're looking here at the very top region, we have a lot of vasculature. Here we have the dorsal vein and the dorsal artery. So on top, we're going to have the superficial dorsal veins. We actually have three sets of dorsal veins, so two on the top that are superficial, one that runs in the center, that's the deep dorsal vein, separated by that subcutaneous layer. Under that, we have the fascia, which is going to separate out the corpus cavernosa from each other and also from the corpus spongiosum. And that's going to look like this in an actual animal. All right, so when I say it's spongy tissue, we can see it really is just going to be a lot of lumen that basically when the blood fills in there, it pools, it's going to cause pressure, which causes the erectile event. All right, do we have any questions on the male reproductive system before I move on to the female reproductive system? I'm going to take that as a no. All right, so the female reproductive system is going to consist of kind of equal and opposite organs. Keep in mind that all of these organs come from the same spot during during embryogenesis, which means that a lot of these guys are going to basically be given different context cues under male or female genetics to differentiate into different organs. So, for example, the testes and the ovaries are a very classic example of paired organs. However, the ovaries don't descend, so the ovaries are going to remain inside the abdominal cavity. They're going to be just directly juxtaposed, but not actually attached to the uterine tube, which is where the site of fertilization is going to occur. That uterine tube is then going to run down into the body of the uterus. Okay, here's the uterus itself. The uterus is going to sit just on top of the urinary bladder, which when you see the fully pregnant female, you'll understand why women have to go all the time, because basically this is sitting directly on top of, all of the weight of the fetus is sitting directly on top of the bladder. But this is normal conditions, non-pregnant conditions. So here we have the uterus, which is connected by the uterosacral ligament to the abdomen. This is here the rectum, which is why this is called the rectouterine pouch, because it's going to be right in between the rectum, right next to the rectum, I guess. Then the uterus is going to be connected to the vagina by a region called the cervix. The cervix has an internal and external os, which is not denoted here, but will be denoted later. The external os is going to be the area that's going to enter into the vagina. Internal is going to enter into the inside of the uterus. And then this is the vagina itself. It's got a lot of rugae. It's going to be able to expand quite large, both for intercourse and also, of course, for the propulsion of a baby through. And then as we get here, we're going to enter into the external organs, and I'll show you that when we get to that, but that's going to include the labia and the mons pubis, etc. All right, so that's a kind of an overview of the female reproductive system. Let's get into it a little bit more. Again, the ovaries are going to be homologous to the testes. Here we go. So that's going to be found here. Here's what an ovary looks like. The ovary is going to be sitting right next to, so just juxtaposed, but not actually connected to that uterine fallopian tube. That fallopian tube is going to descend into the body of the uterus itself. There's a lot of things that are going to connect these reproductive organs to the abdomen, including things like this round ligament, the ovarian ligament, and the uterosacral ligament. That's the one I was looking for. So these are all going to be things that are going to keep these organs in place. Just right next to here, of course, we're going to have the digestive tract. So this here is the sigmoid colon, for example, for going to be entering down into defecation. And then this here, we're going to also have the ileum, which is going to be also part of the digestive tracts. All right, so I digress. Anything else I need to see here? All right, so this is the histology of an actual ovary itself, a frontal section of an ovary, and there's multiple different stages in which an egg can be. So primordial follicle is going to be where all follicles are going to be arrested, basically, during embryogenesis. So in the female, these are all going to be created during the process of embryogenesis and then arrested in meiosis 1 until puberty. From puberty on, one is going to be selected from each ovary, like alternating. So every ovary is going to do it every other month, unless we do it at the same time. Then we end up with fraternal twins, or we could end up with fraternal twins. But I digress. Okay, so the primary follicle, primordial becomes primary, becomes secondary, gets larger and larger. It ends up with this antrum, which is going to have follicular fluid. Eventually, it's going to rupture. When it ruptures, it's going to have around it that corona radiata. Remember, those the cumulus cells we're going to have to penetrate through with that acrosome. Acrosomal enzymes are going to have to get through there in order for the single sperm to get into the zona pellucida and then into the egg itself. After we have rupture of this follicle, this now becomes the corpus luteum. This corpus luteum is going to degenerate, and for the next 2 weeks or so, it's going to send out signals to the uterus that say sustain the endometrium such that we can have an implantation event occur if fertilization does occur. Right? And so that's going to keep that nice endometrial layer thick so that we can actually embed a fertilized oocyte, which is now going to be called, at this point, probably a blastocyst or maybe a morula. It's going to implant. And then at that point, we're going to have a sustaining information sent back to the corpus luteum by HCG that says hang on a little bit longer. If the fertilization event does not occur, the corpus luteum is going to eventually degrade into a corpus albicans, which is going to stop secreting hormones, which means that the menstrual event will occur because the endometrial will be sloughed off, indicating that we did not have a fertilization event occurring. All right, I know that's a lot to take in, but don't worry, we'll talk about the cycles in just a second. Remember, we have the uterine cycle and the ovarian cycle, and they are both in sync, and they're doing two kind of different things. Where are my images? Okay, so this is an actual image of an ovarian cortex. The outside is going to be surrounded by that tunica albuginea. Well, I guess the top layer is called the germinal epithelium. And then we have the tunica albuginea, which is going to be this nice layer here. Here's a primordial follicle, which is then going to become a primary follicle, which then becomes a secondary oocyte. And this takes several months to evolve. Only one is going to go from the secondary to the Graafian to end up being secreted, but it will take many months. So as this one is going to from secondary to Graafian, the primary is going from primary to secondary, if that makes sense. Right, so these are going to have many in a row that are entering into this sequence. All right, the result is the formation of an oocyte. Remember, this is going to be a haploid cell. It's going to hopefully be fertilized by a sperm cell, which is also haploid, resulting in a diploid zygote. And in order for oogenesis to occur, just like we saw in spermatogenesis, we have to have meiosis 1 and meiosis 2 and then maturation. However, when we saw spermatogenesis, we saw that we were going to take one stem cell, turn it into 4 sperm cells. In the case of oogenesis, we're going to create one egg. We're going to take all of the nuclei from what should be the equivalent of 4 cells at the end, put it, not nuclei, I'm sorry, all of the cytoplasm, I apologize, all of the cytoplasm, from what would be the equivalent of 4 cells with a cytoplasm into one. The others are going to become residual polar bodies that are non-functional. And that one oocyte is going to be very, very large with all of the fluid that it needs to be able to sustain the developing fertilized egg should the fertilization event occur. All right, so that looks like this. So here's the oogonium entering into the meiotic event 1, meiosis 1. Now this happens during fetal development. This is where we are arrested upon delivery, basically. Uh, and then we're going to wait about 15 years, 13 years or so, until we enter into our first menses event, or the event of puberty. At this point, we're going to enter into the very first egg being released or ruptured. And typically that actually occurs prior to the first menstrual event. So typically, not necessarily, but it can occur prior to the first menstrual event. So yes, an individual who is sexually active can become pregnant prior to her first menstrual cycle, but probably just the month before. Anyway, my point is that we have a primary oocyte that's going to be arrested here um, at delivery, and then 15 years later, we're going to enter into that secondary oocyte for meiosis 2. Now, meiosis 2 is going to be where we have the polar bodies created, because as I mentioned each time previously when we were talking about sperm, we had a division and another division that resulted in 4 sperm. In this case, we have a division which results in a polar body and a secondary oocyte, and then another division which results in a larger secondary oocyte and extra polar bodies. This oocyte, so again, we end up with 1 oocyte and 3 polar bodies instead of 4 cells. These polar bodies are non-fertile. They're not going to become ova ever. And then anyway, the secondary oocyte is what's going to be released or ruptured, and hopefully we'll end up with fertilization event occurring when it meets a sperm cell. Right, during this time, we're going to enter into the creation of a second polar body, right? And the zygote itself. So the nuclei of the sperm cell and the ova are going to unite, creating this diploid zygote, and then that zygote is what we're going to follow when we look at the process of development. All right, so this is just going to be the images of the primordial follicle entering into the primary, entering into the secondary, finally becoming the Graafian. Don't worry, I'll go back to the beginning. So that's just an image of the secondary and actual TEM. Finally becoming the Graafian follicle, which is that's going to be shed. All right, so I'm going to start you all the way back at the beginning here, which starts us with the primordial follicle. It's going to be surrounded by follicular cells on the outside, and then, so I'm sorry, follicular cells, sorry, that was much larger than I meant to do. Follicular cells are going to be here in purple. And then that whole thing is going to be surrounded by stromal cells. That primordial follicle is going to be arrested in that stage from the point of delivery of the infant all the way on until puberty. During the onset of puberty, we're going to have one turn into that primary follicle and then secondary follicle. From this point, we have the granulosa cells, which are going to become the cumulus cells. Note, we don't have any fluid yet, but we will in the next set. So the secondary follicle is going to start getting that antrum, which is where the fluid's going to be. But I digress. We still have some of the same things that we saw previously, except now those cells on the outside are becoming the thecofolliculi cells. We have a ton of collagen fibers surrounding. We also have inside the granulosa cells. Again, those granulosa cells are going to become the cumulus cells when we actually are going to rupture. All right, so that late primary follicle is going to start to develop that antrum. That antrum is going to end up with follicular fluid. It's going to grow and grow until eventually these cells are going to separate out. So now we're going to have the corona radiata cells, which are actually going to become the cumulus cells, and then all of the rest of the granulosa cells, several of which are going to be left in the ruptured follicles. Some are going to be lost during the transit from there into the ovary. I'm sorry, out of the ovary into the fallopian tract. But some of them, as I mentioned, the ones that are the corona radiata, are going to stay surrounding the oocytes such that the sperm have that last barrier to fertilization. All right, I digress. So that secondary follicle looks like this. Again, here we have the antrum. It's going to be filled with follicular fluid. Then we have, here's the granulosa cells surrounding it, and then the specialty granulosa cells that are going to be the corona radiata cells. These are the ones that are actually going to become the cumulus cells of the egg when it's shed. And inside there, of course, we have the primary oocyte surrounded by the zona pellucida. Now, you may recall the analogy that I gave in class went something like this. You have a tennis ball. The tennis ball is the oocytes. You put the tennis ball inside a nylon stocking. That nylon stocking is the zona pellucida. It's a very thin layer, but it's a pretty strong layer to prevent you from getting into that tennis ball, right? Now, surrounding that, you put a bunch of marbles, and you put all those marbles together by peanut butter. The marbles are the corona radiata cells, and they're held together by a hyaluronic acid, which is basically a glue or a peanut butter. And the sperm are going to have to come up and swim through all of that peanut butter and all of those marbles to be able to get to the zona pellucida, or that nylon stocking. One's going to get through that, and as soon as that occurs, that nylon stocking is going to become an ironclad lock across the entire thing. It's going to become iron instead of nylon. And that means that only one sperm gets through, and then the rest of them are going to be unable to actually fertilize. All right, I digress. That event won't happen until much later, but this is what a secondary follicle looks like. Now we're entering into a mature follicle, a Graafian follicle. This is what is actually going to rupture and then make the journey from the ovary into the uterine tube. All right, so here we have the antrum filled with follicular fluid, again, that corona radiata and the primary oocyte surrounded by the zona pellucida. These granulosal cells are going to be left and some spilled as we have the rupture event, but the ones that will travel with them are the ones that are the corona radiata cells. And again, we are still surrounded by this theca externa and theca internal layer. Oh, goodness, I'm sorry. Here we go. All right, so do we have any questions on the maturation of the actual egg itself or the follicles in the ovary? Oop. All right, so this is a summary of oogenesis and follicular development. Again, we're going to start with the precursor, which is the oogonium. It's going to enter into the primary oocyte and then get stuck at the primary oocyte in prophase 1. So it's going to enter into the very beginning of meiosis, and then it's going to freeze. It's going to freeze that way for 13 to 15 years. Nothing's going to happen until we enter into puberty. At that point, from puberty to menopause, every month a single oocyte will mature. However, it will take several months for the maturation to occur, so we're going to actually have one waiting in the wings for the next one every month, right? This primary oocyte is going to then become a secondary oocyte, creating a first polar body. Sometimes that first polar body splits into two polar bodies. It doesn't always. There's really no need for it to. These are just going to be kind of useless byproducts of this whole scenario. Eventually, the fertilization event occurs where the sperm cell is going to enter into that secondary oocyte. At that point, meiosis 2 is going to be completed. So it doesn't actually get completed until fertilization occurs. And when meiosis 2 occurs, that polar body is going to get popped out of the ova. So previously, that polar body was waiting there to go into meiosis 2. During the end of meiosis 2, we are going to end up with a fertilized ovum, and that polar body is going to get popped out. That's going to result in the 3 polar bodies and 1 ova, which is going to be fertilized. Here's follicular development on the other side. The primordial follicle is going to be inside. Sorry, the oocyte is going to be inside this primordial follicle. And then it's going to become the primary follicle, the secondary follicle, which is going to be here in the primary oocyte, right? Eventually it'll become the mature follicle, which will be the secondary oocyte inside the mature follicle. And the ovulation event is going to result in the ovulated secondary oocyte, which will enter into the oviducts, right, or the uterine tubes, fallopian tubes, right? And that is where the fertilization event will occur. All right, so the uterine tubes, the fallopian tubes are what's going to take that ova in, from the ovaries into the uterus, and that's the particular normal typical site for fertilization to occur. So what does that look like? So here we have the uterine tube. Here we have the ovary. There's the infundibulum. I told you it's not connected. I mean, it's kind of connected, but it's just juxtaposed. Truly, it's a pretty large journey that ruptured oocyte has to get out into the infundibulum of the uterine tube to end up through the ampulla and the isthmus of the uterine tube to enter into the uterus itself. Again, fertilization is going to be occurring here, and then it's going to roll down as a fertilized ova. It's going to be doubling, and so I have 2 cells, 4 cells, 8 cells, etc., until it gets to here. And then the implantation event will occur. The implantation event will occur in the endometrium. So let's look at the uterus itself. We have 3 layers: endometrium, myometrium, and perimetrium. The endometrium is what sloughs off and regrows every month. It's going to be a nice fluid, blood-filled layer for this fertilized, at this point it would be an embryo, right? This for blastocyst, for this blastocyst to implant inside the endometrial lining. Just to kind of show you that there's a lot of things holding this in place, we have one broad ligament that's going to connect the uterus over to the abdomen. We also are going to have the several ligaments that are going to hold the ovaries in place, including the suspensory ligament. It holds the ovaries and the fimbriae of the uterine, holds the uterine tube in place as well. I don't see the uterosacral. Oh, here's the uterosacral ligament here. Now, if we're looking at the cervix, so we're going to again go ovary, out the oviduct into the uterine tubes, from the uterine tubes entering into the uterus, from the uterus entering into the cervix. The cervix is a particular part of the uterus that's going to connect to the vagina. We have the internal and the external os. The internal os is going to be here. The external os is going to be here. This is what we measure when we say a woman is dilated, when she is entering into delivery. We're measuring the size of her cervical opening, basically the size of that external os. And then we are going to enter into the vagina, which, as I mentioned, have a ton of rugae, like we've seen in the stomach, and other distensory organs that need to distend. For example, having a baby passing through. But I digress. So let's talk about the actual implantation event. So once we have a fertilized egg, the fertilized egg is going to, again, the fertilization event occurs here inside the inside the fallopian tubes. And then it's going to enter into the uterus. It's going to help be moved along by these ciliated cells and specific contractions. That's going to look like this. Here's our ciliated cells. And they're basically going to move them along until they find the nicest spots for this to embed inside the uterus. Right? The uterus is known as the womb. It's about the size of a pear. It's going to transport the sperm as well as be involved in the process of menstruation, implantation, etc. And it's going to help develop a fetus during pregnancy and then expel that fetus during labor. It's divided into 3 regions, including the fundus, the body, the isthmus, and the cervix. And it's going to be held by a lot of ligaments, which I just talked about. And this is what it looks like histologically. So here we're going to have 2 different weeks of the menstrual cycle. Here is the 2nd week of the menstrual cycle and the 3rd week of the menstrual cycle. All right, the 3rd week of the menstrual cycle, we're going to have a nice, thick endometrial layer. So the endometrium is split up into the stratum functionalis and the stratum bacillus, right? In week 2, the stratum functionalis is not very large at all. In week 3, the stratum functionalis is nice and thick, ready for an embryo to implant, right? Here's week 2 with the myometrium. Here's week 3 of the myometrium. The myometrium will move down some because we have such a thick endometrium here. Right, now the myometrium is going to be a muscular layer. There's 3 different muscle layers found there. But the endometrium is going to be, again, a tissue that can slough off and regenerate. Now we have 2 different sections. The stratum functionalis, which is going to be shed and sloughed off, and the stratum basalis, which is going to remain throughout the entire monthly cycle and is going to be used to create the new stratum functionalis. Right, so that bacillus is used to create the functionalis. Blood's going to come into the uterus by uterine arteries. There's a ton of uterine vasculature here, lots of different branches, and it's going to be drained, of course, by uterine veins. Some of the major ones, here's the uterine artery, here's the arcuate artery. Again, these are all going to feed into the uterus. If we have the lumen of the uterus, here we're going to be showing the stratum functionalis. You can see it's a nice thick layer. And then this bottom part is the stratum bacillus. So we're looking at the endometrium here, of course. So the stratum bacillus is what's going to remain after and during the menstrual event. This is going to be shed during the menstrual event. And then the stratum bacillus is going to create new stratum functionalis for the following month. All right. So the vaginal mucosa are going to be continuous with the uterus. They're going to run basically through the cervix. So here we have here we have the vagina, here's the cervix, and then here is the uterus. Already shown all of that. So this is basically showing you the mucosal layer. So here we have the inner lumen of the vagina, and then here we have the mucosal layer, which is going to be comprised of non-keratinized, stratified squamous epithelium. Going to look like this. And then we have the lamina propria in this lower level there. All right, so here's the external genitalia of the female. I told you we would get to it. So the external genitalia is going to include the mons pubis, which is going to be a nice fatty region that covers the pelvis so that we don't end up with broken bones during the act of coitus. So this is going to help with padding and cushioning. We also are going to have some pubic hair here, which does a lot of things, including to help um, keep this area nice and lubricated so we're not going to end up with chafing as we walk past, and also going to help with protection from actual impact during coitus. Again, we're going to also have the labia. We have the labia majora, which is the outer layer, the labia minora, that inner layer. At the top of the labia minora, we're going to reach the clitoral hood. Underneath that, we're going to have the clitoris. The clitoral hood here is known as the prepubis. You may have heard of the prepubis earlier when we talked about the foreskin on the male. So this is going to be an analogous organ to the male penis, right? Let's see. We also are going to have the urethral orifice. So this is going to be for urination, and the vaginal orifice, which is where the baby is going to come, and also copulation is going to occur. This is going to be the hymen. The hymen is going to be a nice thin flap of tissue that is sometimes found in females prior to a copulation event. It's pretty thin though, so it can be broken by a lot of things, not just by copulation, including horseback riding or tampon usage, etc. And then we talked about anus, we talked about defecation, when we talked about the digestive tracts. All right, so the female external organs include the mons pubis, which we talked about here. Labia majora, external, labia minora, internal, the clitoris and the vestibule. Orifices, including the external urethral orifice and the vaginal orifice, as well as, and I didn't mention them because they're not shown here, periurethral and vestibular glands, which are going to be involved in secretion of mucus for lubrication as well as some antimicrobial peptides. Here we have what's known as the anal triangle. The anal triangle is going to connect. So basically, your urogenital triangle is going to be on the top, and the anal triangle is going to be on the bottom. They're going to be connected here by this transverse perineal muscle, right? And so basically, we're just looking at the internal musculature here that's holding everything in place to make sure that all of our orifices are going to be lined up. And again, that's going to be the urogenital and the anal triangles. As I mentioned, we have homologous structures in the male and female systems. Some of the really obvious ones include ovary and testes and sperm and egg. But some of the less obvious ones include the labia majora, which would be the equivalent of the scrotum in the male, labia minora, which would be the equivalent of the spongy urethra, and the vestibule, which would become the intermediate urethra, as well as the clitoris, which is going to be similar to the glans penis and corpus cavernosa. And then the glands are going to include the periurethral gland, similar to the prostate, and the vestibular, similar to the bulbourethral. So again, all of these structures came from the same precursor cells in early embryonic development, but were either told to enter into the male differentiation and development cascade because we had a Y chromosome, which has an SRY gene, which causes the male differentiation and development cascade, or we had an XX, which meant we did not have a Y. In the absence of a Y, female is the default sex, and therefore all of those cells were then induced to become these female structures instead. All right, so the perineum is going to be the region that's going to be between, again, the thighs and the buttocks, and that's going to create that triangle that we talked about. So again, the anal and the urogenital triangles are going to make that diamond-shaped area. That whole diamond-shaped area is called the perineum. Now, sometimes this can be severely damaged when women enter into childbirth and delivery, so we might have to have what's called an episiotomy or a surgical incision in the perineum to help prevent terrible tearing or excessive tearing. All right, so this is what the mammary glands look like. So now we're talking about the process of lactation. Mammary glands are actually sweat glands that are specialty sweat glands. So they're modified a little bit to secrete milk and they're going to lie over the pecs. We have specialty cells that are going to be alveoli cells. These are going to secrete milk, and they're clustered in specialty compartments. These guys are going to be found in little lobules within your breast. And we talked about why this is very important for women to take monthly breast exams, because these lobules are generally going to feel very similar every month, but when breast cancer arises, if you find a nodule that you're not familiar with, you really want to know what your baseline layer was, so you can say, no, this really wasn't here 5 months ago, or the last month when I checked. Anyway, I digress. Here we're going to have, again, this is the areola, where we're going to have all of the milk actually coming out. Here we have the alveoli, which are going to be found in lobules. They're going to come out to the secondary tubules, into these mammary ducts, eventually into the lactiferous ducts, and then actually out through the lactiferous sinus, right? And that's what's going to allow you to feed the baby. So we have specialty cells called myoepithelial cells that are going to be surrounding this whole gland, and that's going to help with the expression of milk. The whole point of these glands, obviously, is to synthesize milk and inject milk. That whole process is called lactation. And again, we can have lumps in the breast, which can be either benign or metastatic. Benign one would be something like a fibrocystic disease, which is when we have a breast lump that is going to be either a cyst filled with fluid or just thicker alveoli, but are not actually going to be cancerous. All right, so let's talk about the female reproductive cycle. Again, the female reproductive cycle is going to have an ovarian component and a uterine component. And we're going to have hormonal changes involved that regulate both of these. Remember, it's a cyclical and it's going to interact properly. So we have to make sure that the ovarian and the uterine cycles are lined up properly and timed properly. So that means that we're going to have overlap of the hormones that are going to be involved in the changes that are going to occur throughout the month. Namely, it's going to be GnRH, FSH, and LH. And you're thinking to yourself, weird, I just saw all of these in the male cascade. And that's true. In the process of creating and maintaining testosterone levels, that's true. And that's because, again, we're using the same hormones in both systems. All right, so in the female, FSH stands for follicle stimulating hormone, is actually discovered in the female because we then know that it's just stimulated the follicles. That's going to make the growing follicle get larger and larger in the ovaries until we eventually are going to end up with ovulation event. LH is going to stimulate ovulation as well as the corpus luteum. Remember that corpus luteum is what's going to be left in the ovary after it ruptures, releasing the egg, and that is going to secrete hormones, namely progesterone, estrogen, and relaxin and inhibin. All right, so all of these have their own role. I also might mention that some of these hormones, so GnRH is going to go to the anterior pituitary, so all these are going to be under the regulation of the hypothalamus and the anterior pituitary, namely the levels of FSH and LH. All right, so again, the end result in this pathway are estrogens, progesterones, relaxin, and inhibin. Estrogens are going to help maintain the female structures and secondary sex characteristics, going to help with protein anabolism and cholesterol maintenance, and it's going to inhibit the release of GnRH, FSH, and LH. Remember, we saw that previously too, the end products in the male as well. The end products of the pathway are going to inhibit the initiation of the pathway. All right, progesterone is going to work in concert with estrogens to help create a nice thick endometrium, a nice bloody endometrial layer to help have a nice warm bed for implantation, and also set up the mammary glands to start creating and secreting milk. It also inhibits the release of GnRH and LH, so it's also an inhibitor molecule. Relaxin is going to help inhibit uterine contraction, and it also is going to help stretch out the pubic symphysis, so relaxation and flexibility of the pubic symphysis. Unfortunately for pregnant women, it also helps relax all of the joints, including your ankles and your knees and your hips, while you are carrying a bunch of extra weight around. So it means that sometimes women can get injured by rolling their ankle or twisting their ankle or their knee popping out of socket or whatever. Inhibin is going to help inhibit the release of FSH and LH. So inhibin is going to, again, be an upstream inhibitor of its own creation, if that makes sense. All right, so again, we have two different cycles. The ovarian cycle is everything that's associated with the maturation of the ovum and rupture of that Graafian follicle, and the uterine cycle is known as the menstrual cycle, and this is going to occur in concert with the ovarian cycle, but it's going to directly relate to the endometrial changes that occur in order to prepare for a fertilized ovum or to shed the endometrium to be able to reset the bed for the next month's fertilized ovum. All right, there's a lot in this image. So I'm just going to kind of take it slow. So here's the ovarian cycle and here's the uterine menstrual cycle. And we've kind of talked about each of these independently. In the ovarian cycle, remember, we're going to start with primordial follicles becoming primary follicles, secondary follicles, then mature follicles. Eventually, we have the ovulatory event occur. All of that is under regulation of FSH and LH. So the early follicular phase is under the regulation of FSH, and the maturation phase is under the effect of LH, as is the sustaining corpus luteum, or the choice to degrade the corpus luteum to turn into the corpus albicans. All right, here's the uterine menstrual cycle and how it relates to that. So here we're going to be starting with menstrual day 1. And with menstrual day 1, or 0, I guess, is basically going to be the date of your last menstrual cycle. And that's because it's going to be the easiest way to, or the initiation of menstruation is the easiest way to set an actual date. So we're going to start here with the menstrual phase, which basically is going to be the sloughing off of the endometrial layer. Then the proliferative phase is going to occur while the follicle is developing and maturing. Then the secretory phase is going to continue, again with progesterones and estrogens, nice thick bed, nice layer. Why? Because we have already shed this egg, and hopefully, now that it is in the oviduct, now that it is in the fallopian tube, I apologize, well, now that it is in the fallopian tube, it's going to end up in a fertilization event. Now, a couple days later, after rolling through the fallopian tubes, it's going to implant in the uterus, which will be right around here, and that'll be when we have a nice thick bed for it to implant into. Now, if that does not occur, of course, menstruation will occur because the corpus luteum, which is sustaining this nice thick bed, will eventually become the corpus albicans if HCG is not present. All right, I know that's a lot. Let's look at the different hormones inside the female reproductive cycle when they spike. So the first one to raise itself a little bit is FSH, a follicle stimulating hormone, and that's when we're initiating the onset of the follicle. Then a couple days later, that kind of falls off, although it'll spike again in a moment. But estrogen will kind of start to come up. As estrogen comes up, eventually it's going to be replaced by LH. So as soon as LH turns on, estrogens are going to be turned down a little bit. LH falls, progesterone turns on. Now, progesterone and estrogens are both going to be coming from the corpus luteum, right? So that means that it's basically going to be a spike at the end. And then if we don't end up with a pregnancy event occurring, that progesterone falls off. When that progesterone level falls, because we've switched from corpus luteum to corpus albicans, now all of a sudden, we'll have our menstrual cycle reset again. So this will be the initiation, or back to day 0, right? When we have our menstrual events. All right. So I've touched on everything that I've said here. So please feel free to read that on your own. Female cycles split into 4 phases. First phase is the menstrual phase, which is basically when during this phase when menstruation actually occurs. So it's the shedding of the stratum functionalis of the endometrium, which can discharge what we know as blood, but can also discharge fluid, mucus, and other and tissue, and epithelial cells, etc. It can be pretty thick sometimes. Then we enter into the pre-ovulatory phase, or the proliferative phase, and this is basically when we are resetting or remaking the bed for the implanted embryo. So the endometrium is getting larger, it's proliferating. This happens during just after menstruation and during ovulation, or reaching up to ovulation. And this is going to be a pretty variable time frame. It can last from 6 to 13 days depending. What will happen is our primary follicles are going to develop into the secondary follicles, and a single secondary follicle is going to develop into an ovarian follicle or a mature follicle. All right, and this is actually going to make a bulge. The ovary is actually going to pulse outward. You can actually physically see it, and then it ruptures. Some women are able to actually feel the rupture of this follicle. That's actually going to be known as ovulation. Now, as this happens, the secondary oocyte is actually going to be released into the pelvic cavity, and hopefully, because it's right next to the fallopian tube, it will enter into the fallopian tube. Now, if there's sperm entering into the fallopian tube, the fertilization event can occur. Okay, so this is talking about how estrogen is involved in creating the ovulatory event. So estrogen is going to create a positive feedback on LH and GnRH. LH and GnRH is going to make more LH and GnRH, which is going to cause the rupture That LH surge is going to cause the rupture of this, it's called a corpus hemorrhagium when we actually have the rupture of the follicle, which is going to release that secondary oocyte. Okay, let's hear. All right, so this is talking about the hormonal regulation, both positive and negative hormonal regulation. So red is going to be a negative feedback loop, whereby we have the inhibition of the loop by the downstream created molecule. And then we also are going to have positive feedback loops where we have an increase of that feedback loop. Basically, an increase of something makes more of that something. All right, so if we have moderate levels of estrogens, that's going to help inhibit GnRH, FSH, and LH secretion. Another thing that's going to help inhibit the secretion of FSH and LH is inhibin, right? And that's all going to relate. So here we have the LH and FSH, which are going to be involved in creating the primary into the secondary and the secondary into the Graafian, etc., entering into the process of ovulation. If we're going to increase the secretion of estrogens, that's going to increase the amount of inhibin. The granulosa cells are making more inhibin, and that's then going to have more inhibition of FSH and LH. And that's all going to happen in this first stage. Now, that's going to be when we're basically proliferating our endometrial layer. In this next stage, we're going to be making the endometrial basically prepared for a fertilized ovum. And if that does not occur, then we'll enter into menstruation. Right? And so here we have the same thing happening here in the ovary and the uterus. And we're talking not only about the timing, which means in this case we'll be having the growth of the primary follicles at the same time as the uterus is proliferating. We'd have the ovulation event at the same time as the thick layer is already there. So the endometrium is either end of the preparatory stage or is already prepared. Then the corpus luteum is going to come around and secrete inhibin and progesterone, etc., which is going to help keep this nice and thick if we have a fertilized egg. If we do not, it becomes the corpus albicans and the menstrual cycle occurs. All right, so there's a lot of different hormones that are involved here, including estrogens, GnRH, LH, FSH, relaxin, inhibin, etc. But my point is that this is what helps keep it in a really tight, strict set cycle such that the ovaries and the uterus are on the same page in terms of the timing, such that if a fertilization event does occur, the uterus is prepared to accept it. Okay, so this here talks about what happens with the corpus luteum if fertilization and implantation do not or do occur. So if fertilization does not occur, the corpus luteum is going to degenerate into the albicans. It's called that because it's a white body, so like albino, right? White. And then it's going to stop secreting progesterone and estrogens. However, if fertilization and implantation does occur, the corpus luteum is given information from HCG, which comes from the developing placenta, to maintain its hormone production until the placenta is able to take over. So the corpus luteum will not turn into the corpus albicans immediately after those 2 weeks, but it'll be maintained until the placenta is able to take over the production of creating hormones. All right? So again, the corpus luteum is going to be secreting estrogens and progesterone in order to help support the pregnancy and help prevent the endometrial lining from being shed. Eventually, the placenta is going to take over, and the corpus luteum is going to have a very minor role in the actual sustenance of pregnancy. All right, so this talks about different birth control methods, and there are different failure rates. Really what I need you guys to take from this is that there are some ways that are surgical, including vasectomy and tubal ligation or having your tubes tied. There are some that are going to be non-surgical, like hormonal methods, such as oral contraceptives. We also have non-oral contraceptives, and we also have barrier methods, things like condoms, etc. We have uterine devices, spermicides, multiple different ways, and they basically can work by either stopping the sperm from getting into the vagina, such as a condom, stopping the sperm from getting past the vagina into the cervix, such as a diaphragm, right? Or we also are going to, so those are all going to be types of barrier methods, or we can have chemical methods like spermicides, etc. Or last but not least, we can basically just prevent that from happening by prevent the sperm deposition because of the rhythm method or the symptothermal method, which is when we look at the symptoms of the female, we look at her her cervical mucus, we look at her temperature of the day, and then we find out if she's ovulating. Typically, however, the symptothermal method is used for the purposes of people who want to procreate, so they're trying to find the exact right timing of ovulation, not necessarily for people who are trying not to, but either way, whether you're trying to or trying not to, it helps to know when the woman is ovulating. All right, do I have any questions on the reproductive systems? Nope. So just talking about the development of the reproductive system, which I'm not going to harp on too much, but I do kind of want you to understand that we start with very similar organs, right? The mesonephros, the gonadal ridges, etc. And then these ducts that are these paramesonephric and the mesonephric ducts, which eventually are going to get information either from the Y, which initiates male development, or the lack of Y, which triggers the default cascade, which is female development. And that's how the testes are going to form or the ovaries are going to form from those gonadal ridges, right? So the urogenital sinus is going to turn into here the prostatic utricle, which is eventually going to become either the uterus, or in this case here, the prostate there, right? So depending on what clues we're getting from our genetics, which then are going to tell us the male differentiation cascade or the default female differentiation cascade, we will take the same precursor cells and turn them into either male or female reproductive organs, right? All of the external genitalia are going to develop from the genital tubercle region. Right, again, this is kind of the same thing. We start with the undifferentiated stage as a 5-week embryo. We're either going to get the male differentiation or development initiation or not. If we do, then this genital tubercle is going to become the penis. If we don't, it becomes the clitoris. Again, if we do, the uterine-urethral folds become here the labral scrotal swelling and then eventually become the scrotum. But in the female, that will become the perineum and eventually become the vaginal orifice, right? All right, so as we get older, males and females are affected differently. Now, while males do have a decline in reproductive health, they can still fertilize eggs well into their 90s, up until their deathbed. Ladies, sometime around 40 and 50, the ovaries are going to stop responding to the stimulation of gonadic hormones, which means their follicles no longer are going to enter into follicular development. That's going to be called menopause. And menopause will be the last menses, or the pause of your menstrual cycle. And unfortunately, you don't necessarily ever know when it's going to be the last one when you enter into this time frame, but basically what that means is that you are past the age where you were fertile enough for a fertilization event to occur because you are unlikely to be ovulating. Some of the symptoms of menopause are going to include hot flashes, sweating, vaginal dryness, headache. You can also have some depressive events that can occur, and we can have fluctuations in weight because our hormonal changes are going off the chart. All of these are able to be modified or treated with supplements. So it's pretty easy to get some hormonal supplements. You also have emotional fluctuations that can occur again because of the hormones that are kind of going a little bit wonky. Additionally, post-menopause, women can increase their rate of osteoporosis, uterine cancer, and breast cancer. So there are certain things that they now are going to come into a higher risk category than they were in prior to menopause. Now, again, as I mentioned, men have declining reproduction, but there's not an actual termination date. Males can often retain reproduction until their 80s and 90s, although they do end up with lower levels of testosterone, which can also decrease the amount of viable sperm. Right. All right, so I think that covers everything I wanted to talk to you guys about. All right, so do we have any questions on any of reproduction before we get into inheritance and development? I'm going to take that as a no, and I'm going to move on. All right, so I don't know what happened here to the outline, but I didn't have the chance to fix it. I just noticed it last night. So inheritance got kind of wonky. It's not A, B, C, D, but just bear with me. It's bullet points. Basically, inheritance is when genetic material is passed from parent to offspring from multi-generations, right? And genetics is how we study the inheritance of genes. Now, we have 23 pairs of chromosomes. That means we have 46. In the human, that's going to be 2N is 46, 1N is 23. So if we're looking at an egg or a sperm, we're looking at N, or 23 in the human. If we're looking at every other cell in the body, we're looking at 2N, or 46. Genes can be homologous. That means they can either be the same or they can be different. So if we have genes that are different, they're called allelic variations. So if they're found in the same location, but one's blue eyes and one's brown eyes, for example, those are called alleles. And because we have two genes for each, we have two alleles for each of our traits, because we have one maternal and one paternal gene, we can either be what we call heterozygous or homozygous for that gene, meaning we can have two of the same blue eye color, for example, or two different, one blue and one brown eye color. Right? And if we end up with a mutation that gets passed on, that's called an allele. Again, it's a heritable change in the gene itself. Let's talk about Punnett squares for a second. Punnett squares are how we look at the genetics from each of the parents. All right, so this is looking at sperm types and egg types. As I mentioned, sperm are going to be N, or just half, and the actual parents, mom and dad, are going to be 2N, or diploid. So these are diploid cells that can give rise to haploid cells. So that means that we can have two different options of sperm type from a father that is heterozygous. All right, so this individual is going to be a uppercase P and a lowercase p, and this one again, uppercase P and lowercase p. So both mom and dad can create two different types of gametes. So two different types of sperm and two different types of eggs. So if we separate that out in what this is called a Punnett square, we basically are going to align the sperm types on this side and the egg types on that side and then bring them together. In this box, we'll bring these two. In this box, we will bring these two. In this box, we'll bring these two. And in this box, we'll bring those two. And what we're showing here is that we're going to get one individual that has two uppercase P's, two individuals that are going to get one uppercase and one lowercase p, and one individual that is going to be two lowercase p's. I'm going to define this in the terms of what we would be calling about genetics. So, to backtrack, the uppercase P in this case is considered dominant, while this one is considered recessive. All right? Now again, if we have two of the same, that's considered homozygous. So this would be considered two of the same dominant, so homozygous dominant, and this would be two of the same recessive, so homozygous recessive. We would then also have two offspring, which will be what we call heterozygous. So this also brings me to another point. We can look at the genotype or the phenotype. The genotype will be looking at, again, homozygous dominant, homozygous recessive. However, because these two will have similar phenotype, in this case we're looking at PKU, but we could be looking at flower color, etc., because this dominant gene is going to present itself the same whether we have two copies or one copy, all three of these are going to appear very similar phenotypically, or have a very similar outward appearance, in that these guys do not have the disorder and this one does. So while we have a genotypic ratio of 1 to 2 to 1, right, 1 to 2 to 1, we have a phenotypic ratio of 3 to 1. Right? I do hope that makes sense. Anyway, that's how we can use a Punnett square to look at the possible offspring that can come from different genotypes. Again, the genotype is the genetic makeup of a trait. The phenotype is the outward appearance that is related to that genetic makeup. We can have a dominant allele that's going to dominate or mask the presence of another allele. That one's going to be represented as an uppercase letter. The allele whose presence is masked by the recessive, mask is called the recessive, I apologize, and that's going to be a lowercase letter. And just to be clear, they are both going to be the same letter. So if we're looking at purple and white, purple is dominant to white. That means purple is going to be uppercase P. White flowers, for example, purple flowers are dominant, white flowers are not recessive, and basically that's because purple is going to make pigment, so pigment will be dominant over no pigment. But my point is, white is not denoted lowercase w, it's denoted lowercase p, because it goes along with whatever the purple is, right? Now, we do have other types of things like incomplete dominance or codominance. Incomplete dominance would be when we do have a little bit of the traits. We have kind of a push-pull or an intermediate trait. And then we also have codominance, which basically would be that they were both dominant. One good example of that would be the ABO blood type. So for example, A and B are codominant. Basically, they do create a sugar. They just create the A sugar or the B sugar. but both of them are dominant to O. So if we have AO, you will appear to be A. If you have BO, you will appear to be B. If you have AB, then you'll have both sugars on your surface, right? Only if you have two O's will you appear to be O. All right, so we also have codominance. And from here, we can determine, based on what the parents are, what the appropriate options of the offspring are. So if, for example, both parents are A, now again, because they're genotypically A, or phenotypically A, we don't know if this individual is A, comma A, or A, comma O, because it would be masked. Same here, which means that they could both be A, comma O. If they're both A, comma O, the offspring could be O. But if they're not, then the offspring will probably be A. So depending on what your parents are, you can end up with different offspring. I'm not going to walk you through each of these. I did that in the lecture if you would like to. But my point is that we have codominance. If we have A and B together, it's called AB. But if we have A and O together, it's just called O because A is dominant to O, right? We can also have polygenic inheritance, which basically means that we have the combined effect of multiple genes. And then we can also have multiple different effects and environmental factors. For example, your skin color changes when you go out into the sunshine. That's an environmental factor that could be taken into account when you're looking at the skin tone of someone. So here's my point is that when we have multiple different genes that result, so here we have 3 different genes, gene A, B, and C. And for each of them, we can either have capital A or lowercase a, capital B or lowercase b. And if we have the capital, it's going to be very dark. And if we have the lowercase, it's going to be very light. So if we have all 3 capital, very dark, all 3 of them are going to be homozygous for the very lowercase, that's going to be very light. You can also have any intermediate phenotype whereby we have 1 capital A and 1 middle A. So basically, if we start at the P generation where we're very dark and we have 3 homozygous for 3 dark genes, A, B, and C, and we cross that with someone who's homozygous for 3 very light genes, we're going to end up with the offspring being an intermediate phenotype that's approximately in the middle. Because each of them are going to have 1 dominant and 1 recessive allele for A, for B, and for C, right? So you can see 1 capital and 1 lowercase for A, B, and C. And then when we cross these guys together, we all of a sudden get an entire range of offspring. Because the sperm can range from being all 3 lowercase to all 3 uppercase. And then it can be any combination of the 2 uppercase and 1 lowercase, or any combination of the 2 lowercase and 1 uppercase. So that gives us a total of 1, 2, 3, 4, 5, 6, 7, 8. So 8 on this side and 8 on this side gives us 64 different combinations between very, very light, namely all 6 are going to be the lowercase, to very, very dark, when all 6 are the uppercase, right? So we can have a very wide range of phenotypes that come out of the offspring if it's a multigenic trait, meaning that there's many genes that are involved in its creation. I've already told you this, but the human chromosomes, there's 46. That's called 2N. 23 would be N. We have 23 pairs. 22 of them are called autosomes. That means that they are not sex chromosomes. And then 1 pair is the XY. If we were to look at all of the chromosomes, we would call that a karyotype. It starts with the largest one is 1, the smallest one is 22, and XY is this weird little odd couple out whereby X and Y pair, but Y only has on it the male differentiation and development cascade, whereas X has almost everything else. It is going to have, for example, blood clotting, so it can be related to hemophilia. It has color vision, so it can be related to color blindness. It has male pattern baldness, or etc., widow's peak. So there's a lot of different things that are found here on this X, whereas the Y only has very single-minded vision, create male offspring. But the rest of them are going to pair very nicely. So these are all called autosomes, and these are our sex chromosomes. And again, in XY, this individual is going to be a female. I'm sorry, this individual is going to be a male. I apologize. XY is going to be a male. A female will have 2 X's. So an individual with XX is a female. An individual with an XY is a male. And interestingly enough, a female can only give rise to an egg that is X. So that means that all eggs are going to be X bearing. Why? Because females are XX. She does not have a Y to pass on. So all eggs are X. That means that the fertilization event is either going to have an X bearing sperm or a Y bearing sperm. And that is what is going to determine the gender of the offspring. The sperm itself that is selected, or that actually makes it through triumphantly to fertilize the egg. That means that Henry VIII, for example, who had several of his wives murdered because he was not getting male offspring, really had no one to blame but himself in the end. Again, there are several things that are found on the X chromosome. That means that we have some things that are sex-linked traits, such as red-green color blindness and male pattern baldness. These are more likely to appear in males because males only have one copy of an X, meaning that they're what we call hemizygous instead of um, homo or heterozygous. That means they only have one copy, and basically what you get is what you get, and you don't have any choice in that matter. So if you get one that has male pattern baldness, then so be it. Whereas women, we'd have to have two bad copies of the X, because even one good copy would be enough to sustain us. All right, so that's inheritance. I'm ready to move on to development. If you are, you guys ready? Any questions on inheritance? All right, I'm moving on to development. All right, so development is basically what happens from the fertilization event to delivery of a infant. And the first couple of months following fertilization is going to be called embryonic development. And then after the placenta takes over, it's called fetal development. All right. Up until the fetus and up until the placenta takes over, it's considered an embryo. After embryo, it's considered a fetus. Now, the entire process is called embryology, or not the entire process, the early stage. So, watching the development of the embryo is called embryology. And that's the study of the development from fertilization through all of the embryonic stages into the fetal periods. Now, in medicine, there's also obstetrics, which is basically the OB part. Obstetrics and gynecology would be OB/GYN, and they're put together. And obstetrics is basically the medicinal branch that's going to deal with pregnancy, labor, and the neonatal period, which is basically just after the baby is born. Now, the prenatal development is broken up into 3 major trimesters. And that's going to basically be the first 3 months, the second 3 months, and the third 3 months. Now, the embryonic period is going to go from fertilization. Remember, the fertilization event is going to occur in the oviducts. I'm sorry, if I keep saying that. It's going to occur in the fallopian tubes. And then from there, it's going to travel along the fallopian tubes into the uterus. And that is where we're going to have the site of implantation. And this is what the actual fertilization event looks like. So here's the oocyte in pink and the sperm cell in blue. The sperm head is going to get in, the neck with the mitochondria, and the tail is going to be left on the outside. And the zona pellucida, which right now is a nice, warm, inviting layer where for the sperm to bind, will immediately become an ironclad fortress where no second sperm is able to get through. All right, so this is basically the path that the sperm cell have to take. So just to kind of go back to my previous analogy, this is the tennis ball covered in a nylon stocking, covered in marbles that are held together with peanut butter. And the sperm have to get through those marbles, and in order to do that, they have to digest the peanut butter. And to do that, they have to release these acrosomal enzymes that are found on the tip or the head of the acrosome. And once the very lucky sperm makes it through all of the cumulus cells, then they're going to enter into, so the cumulus cells are like corona radiata that we're talking about out here. And then we enter into the binding of the zona pellucida and penetration of the head into the plasma membrane of the secondary oocytes. At that point, the polar body is going to be shed, and we're going to enter into the very end stage of meiosis II. So that's going to complete the second meiotic event. All right, again, the zona pellucida region has a ZP binding site called, or ZP binding protein called ZP3. It's a sperm receptor. Basically, it's going to trigger the acrosomal reaction, which is releasing the acrosomal enzymes, which are going to be our peanut butter eaters that get us through these cumulus cell layers such that we can get to the plasma membrane and have the fusion of the sperm with the egg. That's called syngamy. Now, syngamy is going to be an event that occurs only once. A second sperm cannot happen. Why? Because there's severe chemical changes that are going to be called a block to polyspermy. So a second sperm cannot get into the oocyte. Basically, that nylon membrane turns into an iron chastity belt. All right, so once the sperm enters into the secondary oocyte, then meiotic event is completed. So meiosis II is going to be completed. Male pronuclei and the female pronuclei fuse. So here's the 2 pronuclei about to become one. And then at that point, then we are going to have what's called a zygote. Okay? We can also, this is talking about how twins are created. Dizygotic twins are called fraternal twins. And basically what happens is accidentally or erroneously, two different ova are released. Either they're both released from the same ovary, which is rare, or both ovaries are induced to release an ova at the same time when they ought to be on alternating schedules. So basically, two ova and two sperm are going to fertilize. We have two separate fertilization events. This is essentially going to be siblings that are carried in the same womb at the same time, right? So they're going to be genetically dissimilar as any other siblings. Now, we also have what's called monozygotic twins. These are identical twins. These are going to come from a single fertilized ovum that at some point in the splitting into two, and then into four, then into eight, and then into 16, a cell or two falls off, but the differentiation stages haven't occurred yet. So these are basically just embryonic stem cells, and so they are basically going to become a whole new organism early in development. They're going to contain identical genetic material and are always going to be the same sex, whereas fraternal twins could potentially be one male and one female. All right, so the zygote is going to be that fertilized embryo. It's going to enter into the 2-cell stage. This is our first day. Then it's going to cleave into 4 cells, and then into 8, into 16, etc. But I want to make clear here is that we're not going to increase in size in this early stage. We have a very large egg. It's actually going to be the largest cell in the human, one of the largest cells in the human body, and it's going to then cleave into smaller and smaller cells such that the overall size of the zygote remains the same as it turns into a morula at day 4 and a blastocyst at day 5. And eventually that blastocyst is going to end up binding to the endometrium of the uterus. Now, mind you, the blastocyst is hollow inside, so you can't really see that, but it's going to be binding to the endometrium of the uterus, and it's going to bury its way into the uterus until it's finally entirely embedded into that nice thick endometrial layer, which is present at this time. Okay, so here we have the actual cross section or frontal section through the blastocyst and the endometrium of the uterus. The endometrium of the uterus has endometrial glands. They're going to be secreting uterine milk. We also have a lot of vasculature. Remember, this endometrium is nice and thick with vasculature tissue. Here's the trophoblast set of cells. The blastocyst cavity is going to be inside. It's going to be kind of a hollow opening. And these guys are going to be the embryoblasts. This is the inner cell mass, which is quickly going to start differentiating into the embryo. Oh, after implantation, the endometrium is now called the decidua. So instead of being called the endometria, it's now called the decidua. It's called the decidua basalis, decidua capsularis, and decidua paritalis. And that looks like here. Right, so decidua capsularis is going to be the region that sits right on top of the implanted blastocyst. The decidua basalis is what's going to surround it, and the decidua paritalis is going to be the outermost layer that lies just adjacent to the capsularis. So again, the capsularis is only going to be the part that covers the actual blastocyst. So this is basically everything that occurs in the first week after fertilization. So the ovulation event occurs. Here's the ovary. Mind you, the fertilization event is going to occur in the uterine tube. That means the sperm are going to have already swum all the way up through the vagina, through the external and internal areas of the cervix, through the uterus, into the fallopian tubes. Again, fertilization is going to occur here about 12 hours after the ovulation event. Then we're going to have a cleavage event occurring. The first one's going to happen about 30 hours post-fertilization. And then we're going to get another one, 24, etc., until we eventually are going to end up with a blastocyst. So the morula is going to be what drops into the uterus, but the blastocyst is what's going to be what implants. And the implantation event, again, is going to occur in the endometrium, which is very quickly after going to be called the decidua. All right? So the endometrium is non-implanted, and the implanted endometrium then becomes the decidua. Now, as I mentioned, there is a lot of space here between the ovary and this fallopian tube, and sometimes something called an ectopic pregnancy can occur. Basically, an embryo or a fetus can develop outside of the uterine cavity, somewhere in the abdomen. Oftentimes this occurs in the fallopian tube, but sometimes it can occur in the ovaries, right, if it doesn't quite rupture and the sperm get there. In the abdomen, in the cervix, or in the ligaments. Sometimes ectopic pregnancies can be very severe and can be life-threatening. Oftentimes they will result, particularly if they result in implantation inside the uterine tube, it will result in a removal of that fallopian tube. All right, so let's talk about what happens to the trophoblast. The trophoblast is going to turn into a syncytotrophoblast um, and a cytotrophoblast. And that looks like, here we go. So the trophoblast layer is that external layer, and it's going to become, here's the um, cytotrophoblast here and the syncytotrophoblast there. Those are going to turn into the fetal associated tissues, such as the placenta. And then inside, we're going to be creating a bilaminar disc. Now, it's only bilaminar for a short while. Very soon, it's going to become trilaminar as we have the intravasion, basically, of the mesoderm into what is currently, it's the hypoblast and the epiblast, which are the precursors or the embryonic ecto and endoderms. Okay, this whole region here is going to become the yolk sac. The yolk sac is what's going to feed the developing embryo up until the placenta takes over, right? Surrounding here we have lacunae. Lacunae are going to be these large areas in the decidual layer where we're going to be able to eventually pool with blood. This is going to be a precursor to the placenta where we're going to have nutrient exchange occurring again. And we don't have that yet, so the yolk sac is going to be what we're depending on to nourish this embryo at this point. All right, so here's our blastocyst. Again, we're going to have our epi and our hypoblast. Sorry, epi in blue, hypo in yellow. We also have amniotic cavity. It's going to be filled with amniotic fluid. Here's our amnion. And then here's our yolk sac. Again, the yolk sac is going to help feed or nourish the developing embryo. Around here we have the lacunae. We've seen lacunae before when we talked about the bone. Lacunae just means openings, right, or channels. And then we have these regions here, which are going to be the maternal sinusoids. And that's going to basically be an area where we're going to start eventually pooling blood from the maternal tissue around, although there will be a membranous barrier, around vasculature of the fetal tissue so that it's able to uptake things from the maternal blood supply. All right, we touched on that. On this. Oh, so gastrulation is going to be the very first major step that's going to occur here, taking that two-dimensional bilaminar disc, making it a three-dimensional trilaminar. I'm sorry, it's still two-dimensional. It's going to undergo neurulation. So gastrulation still two-dimensional bi to two-dimensional tri. That's gastrulation. Then neurulation will take that three-layer trilaminar disc and make it three-dimensional. Right? So gastrulation is basically going to be when we're developing what's called the primitive streak. So we're basically going to be making the head end and the tail end. We're going to be basically giving ourselves context clues, so cells are going to align along each other so that they know where they're going to end up in the end result of the embryo. Here's our oropharyngeal membrane. This is where we're going to have our mouth. Here's the tail end. This is where we're going to eventually have the anus. All right, so that bilaminar disc is going to become a trilaminar disc by the basically we're going to have invasion of this mesoderm right on in between the developing endo and ectoderm. So what was previously the epiblast and the hypoblast are now going to become the ectoderm and the endoderm, and the mesoderm is going to come right on in, and now we're going to end up with a trilaminar embryonic disc. This is about 16 days post-fertilization. All right, this I'm not going to go over. Sorry, the diagram I will, but this table I'm not going to, but basically this is letting you know which of the adult structures are going to be produced by the endoderm, the mesoderm, and the ectoderm. So which of these layers is going to become what tissue? And basically, as a rule of thumb, ecto becomes outside, endo becomes inside, and meso becomes middle, like musculature, etc. But you can go ahead and take a look here at this table to figure that out. All right, so the next thing that's going to happen is the process of neurulation, where we're going to create the notochord. The notochord is going to induce the neurulative pathways. So once the notochord is created, the notochord is going to induce cells to form the neural plate. That looks like this. Okay, so here's our neural plate, head end and tail end. We're going to end up, here's our primitive streak at 17 days. Here's our primitive streak at 19 days. It's going to get more and more complex. Somewhere around 22 days, it's going to actually completely invaginate, creating that neural folds. And we're going to start seeing the appearance of somites. If we do a cross-section through there, we will see here that at day 17, we're getting that mesoderm. Here's our notochord. Here's our neural plate. That neural plate is then going to turn into a neural fold. That neural fold is going to then eventually turn into a neural groove where we have neural crest cells on the top. The neural crest cells are then going to turn into an entirely separate layer on top, and that notochord is going to become the spinal cord and spinal column and associated structures. Okay, so eventually that neural plate is going to make the neural folds and the neural groove to form what's called the neural tube. And the neural tube is going to become the spinal column. All right, so this is again showing what that looks like. Here's that neural plate. Here's the notochord. The neural plate is going to start folding. These here are going to be the future neural crests. So these are basically going to be coming together as this becomes the circle. All right. The neural folds are going to invaginate inward until that neural groove is going to become a neural tube. And the neural crest cells here are going to come together to sit on top of the neural tubes, right? They sit in the crest on top of it. All right. So basically that's going to create our neural crest and our neural tube. Again, the neural tube and the notochord are going to become our spinal cord and spinal column. Now let's talk about the brain for a second. This is the developing brain. Here's the 3 to 4 week embryo has just a couple of the primary vesicles, namely the procephalon, the mesencephalon, and the rhombencephalon. So the prosencephalon is going to be the forebrain. We're going to have the developing eye. The mesencephalon is the midbrain. Here, the rhombencephalon, the hindbrain, where we're going to have the otic plate of the developing ear. Later on, we're going to have some secondary vesicles developing, including the telencephalon, diencephalon, metencephalon, and myelencephalon. All right, and they're going to look like this. Again, we're going to have the forebrain here, the diencephalon and the telencephalon are going to be the forebrain. Then the midbrain is going to have the mesencephalon. And then that hindbrain, so the rhombencephalon, is going to turn into the metencephalon and the myelencephalon. Right, and now we're also going to end up with a little bit more precise developing eye and developing ear at the 7-week mark. Okay, somites we talked about, we showed those in red. Those are going to become the, they're going to become the, I can't come up with this word, vertebrae, the spinal vertebrae. They're going to surround the spinal column, going to be part of the developing nervous system. And this is how we can look at an embryo and determine how far along in embryological development that embryo might be, even if it's a little bit larger or smaller than other embryos its age. All right, so let's talk about the cardiovascular system. So vasculature arises from what starts off as blood islands with a small little lumen in between, which eventually is going to have these get filled in by these angioblast cells. Angioblasts are precursors or angiogenic tissue, which will eventually become these lumen lining cells and the blood cells themselves. So this is what's going to help separate out. Now we're going to have vasculature as a blood vessel here. Right, the angioblasts, again, they're going to form the walls of the blood vessel. Let's talk about the heart and how the heart is created. The heart is going to start off with 2 endocardial tubes. Those endocardial tubes are going to fuse in multiple spaces and become primitive ventricles and primitive atria, as well as this tricus arteriosus, which is going to become the aorta, and the sinus venosus, which is going to become the superior and inferior vena cava. So we're going to have a folding event that occurs. It's going to take that primitive ventricle and primitive atria and make 2. But keep in mind here that we're going to actually have, there's going to be leakage between our atria such that we can allow a little bit of backflow. That hole will eventually be closed. Also keep in mind that we are going to have 4 different branches off of the aorta, but eventually in the adult we're only going to have 3, so one of those branches is going to become a ligament. But we talked about that when we talked about the hearts. Let's see. So let's talk about the placenta. So basically the placenta is going to come from the chorionic villi. So the chorionic villi are going to start as small little projections of the cytotrophoblast layer that are going to have these capillary bed that are blood filled, right? These chorionic villi. See all these lacunae. Remember we saw lacunae? We saw that also in the bone. It just meant like kind of holes or spaces. So this is going to be spaces that are going to be able to fill with blood. Here's our maternal sinusoid, which is going to be connected to these chorionic villi. Right? It's going to pool with blood, and it's then going to eventually be a spot where blood can cross into the fetal blood supply. So here we're looking at the chorionic villi. So this is going to be the fetal blood supply. Here's the maternal sinusoid. The chorion itself is going to be composed of the cytotrophoblast and the syncytiotrophoblasts, which are eventually going to become the placental layer itself, or the fetal side of the fetal associated tissues of the placental layer. We also have the intervillous space and the stalks that are going to connect the maternal layer to the fetal layer. At this point, remember the embryo is still relying on the yolk sac for nutrients and nourishment. The placenta is developing, but is not yet going to be what is going to be sustaining this embryo. The yolk sac is just going to get smaller and smaller and smaller until all the nutrients are absorbed, and that'll happen right around the same time that the placenta is capable of taking over. All right. So again, here's the chorionic villi. They're now becoming the placenta. So here we have the umbilical cord, which is going to be the fetal vasculature. It's going to have the umbilical vein and the umbilical arteries that are going to feed into these regions here, which are the chorionic villi. Again, that's the fetal placenta portion. Here's the maternal portion, which is going to be in the decidua basalis of the uterus, right? Remember, implantation is going to go all the way into what used to be the endometrium, but is now the decidual layer. And it's going to pool with blood. So we're going to have maternal vasculature here that's going to then leak across this membrane to be able to be picked up by the fetal vasculature. The purpose of the placenta is to basically allow for nutrients and oxygen to diffuse out of mom's blood supply into the fetal blood supply, and then all of the waste and carbon dioxide from the fetal blood supply can diffuse out into the maternal blood to be off-gassed or released from the body. So this is what a placenta looks like. This is the fetal surface of the placenta. Here's the umbilical cord. We have plasticized the arteries to show you that we have large umbilical arteries and large umbilical veins. This is considered fetal associated tissue. It's not maternal tissue, although it is a temporary organ that is created in the female. We do have maternal tissue in there as well, but with the fetal surface is basically going to be fetal associated tissue. And this is actually going to be considered the afterbirth. So after the baby is born, the placenta will detach from the uterus, and they will also have extra pushes, etc., where the placenta will then expel from the body. And oftentimes this takes several hours to occur, so a woman who has never been pregnant before might be surprised because she might think the entire event is over. She's had her baby, and then all of a sudden she has additional contractions and expels this very large blood-filled organ. It must have been quite shocking if you don't know what to expect. So the placenta is a very important organ, and the way that it is going to be implanted into the uterus is going to be really important in terms of how high or low risk your pregnancy is. There's a condition where it's called placenta previa, where the placenta is going to become implanted in the wrong part of the uterus, basically near the external or internal os, sorry, near the internal os of the cervix. And so if this is the case, they're probably not going to have vaginal delivery. They're probably going to induce a cesarean section. And then you can also have a condition where the placenta actually pulls off of the decidua or becomes a little bit detached. And that would be something, I think that's called placenta abreva. And that's going to be something where you could have that, you'd end up on bed rest, etc. All right, so I digress. So let's go back to week 4 of development when we are going to be converting the embryo from a two-dimensional trilaminar embryonic disc into a three-dimensional cylinder, right? So the first thing that happens is neurulation, right? So once we have the neurulation event occurring, now we're going to end up with the neural tube and the notochord. We're also going to, at the 24 days or so, have these lateral folds coming down. The lateral folds will then wrap all the way around. At this point, we're going to be losing the yolk sac. The yolk sac is going to get smaller and smaller as we're using up the nutrients from it, until it is eventually going to leave just a little bit residual, which is called the midgut. It will then become the digestive tract, right? So this region, which has nourished us even in embryology, will continue to be the region that allows us to be nourished as we become infants and then adults. All right, so we are going to develop these are called pharyngeal arches, and in between them we're going to have pharyngeal pouches, right? These are going to be somites. I'm sorry, in between them we have pharyngeal clefts. The pharyngeal pouches are what are going to hold the pharyngeal arches. This is going to be the otic plate coat and the lens plate coat are basically the precursor to the eye and the ear. We also have here the esophagus, so the midgut, the early,
I'm sorry, the early breathing tube, not the gut. So the early breathing tube, although of course at this point we're not actually breathing, right? We're in amniotic fluid and all of our oxygen exchange is occurring through the placenta. So no air is actually coming in here at this point, but we're setting the stage for that to happen, also for food to be happening. So we will be swallowing through our esophagus and breathing in through this trachea, which is going to be this little lung bud right here coming through. So this is all going to come from that precursor to the gut. Here's going to be a section through the pharynx. So we're going to see our pharyngeal arches. These pharyngeal arches, of course, are going to end up becoming parts of the throat. And we are going to have muscle layers, nervous layer, and cartilaginous layer. And we're going to end up with 3 layers of, and that's going to include that cartilage layer that surrounds our throat, which can help our throat from collapsing. All right, the developing ear is going to develop from the otic placode. And at this point, we're going to have limb buds. This is 4 weeks. We have our otic placode. We have limb buds, but no hand buds, no finger buds. We still have a tail. The tail is going to disappear. So the tail is obviously not going to be something that stays with us until infancy. This here is a heart prominence or a pouch where the heart is going to be developing. Remember, the heart's going to have to fold a couple of times, so it's going to need a lot of space to do that. And then here's a liver prominence, which is going to be an area where the developing liver is going to be taking place. Now at the 6-week embryo stage, we've now taken that limb bud and made a hand plate. Again, we don't have any fingers, but we have hand and foot plates that are going to be pouching off. The eye is going to look a lot more familiar. Again, that ear is still here. Our tail is still here, but we will be losing that shortly. And these are those pharyngeal arches that we talked about that are going to become the throat. Here's our 7-week embryo. Now our eye is becoming a little bit more familiar as an eye. Here's our hand. Again, we don't quite have fingers yet, but it's going to become more clear that it's a hand and not just a hand bud. Same thing with the foots. So you're starting to see things that are going to be recognizable as a human or an infant. Let's see. So here we are at week 8. Come on. At this point, we have formed digits and we have our eyelids. So we're actually starting to have, this is cartilage, not bone, so no ossification is happening, but we're starting to get protective measures over our important internal organs, which are forming, such as our precursors to our ribs. We're also starting to get precursors to some of our other bones, some of our long bones, like our humerus here, as well as our scapula. All right, so this is basically a summary of all of the things that are going to happen during embryonic and fetal development. This basically shows you what happens from 20, 24, etc. as we get older until we finally end up at a 26-week fetus when we actually can recognize this as a human. To be clear, these stages are going to occur very similarly in a lot of mammals. So until a certain stage of development, you truly wouldn't be able to tell off the bat whether or not you were dealing with a mouse embryo, an elephant embryo, or a human embryo, unless you happen to be an embryologist and knew specifically. But at a certain point, we do end up looking very distinct. Right now, we have a very human embryo shape here around the 13-week mark. All right, so this table summarizes all the changes that occur during embryonic and fetal development. I am not going to linger on this too long because I have another review session to get to in about 45 minutes. This is going to be how large the developing infant, developing embryo, and then fetus is going to be relative. Obviously, this is not depicted in inches, but just so you can kind of see the shape. Originally, the head is going to take up about 2/3. I'm sorry, about 1/3, I apologize. About 1/3 of the body, and then later on it's going to be much smaller in proportion. So really the head and the developing brain is going to be the first thing in development that we really are going to focus on during embryogenesis. All right, so teratogens are anything that can cause a problem during embryonic development. Typically, these are not going to be chemicals that are going to be harmful to adult humans, but are going to be something that can cause issues to a developing human during embryogenesis. Now, some of these teratogens are going to include things like alcohol, but they can also include pesticides, industrial chemicals, hormones, antibiotics, prescription drugs, etc. That's why pregnant women have to be very, very careful what they put into their body. And obviously that means quitting drinking and quitting smoking because those are known teratogens, although they are also known carcinogens, at least cigarette smoke is. But carcinogen and teratogen are two different definitions. A carcinogen is something that causes cancer in adults or in children when full in formed humans, and a teratogen is something that causes developmental defects during embryogenesis. All right, so this last little bit talks about how we can check on a fetus or an embryo And we can do that a couple of ways. We can use an ultrasound. So we've all heard of an ultrasound. Basically, it's a sonogram, and we use sound vibrating down and reflecting back to determine um, the age of the fetus and the size of the fetus and viability and growth, etc. We can also determine which direction the fetus is in, making sure that it's flipped head down at the right time, etc. We can determine if we have multiple pregnancies because we can see more than one in there, or different fetal maternal abnormalities. Some other things that we can do that are a little bit more invasive are going to include testing that we get of the amniotic fluid. We'll get that by way of amniocentesis. Basically, what happens is we take a long needle, we stick it through the abdomen to withdraw amniotic fluid. And inside there's some cells that have sloughed off. So we're going to have some fetal cells, and they can take a look at those fetal cells and discover whether or not that individual is suffering from a certain genetic disorder, such as Down syndrome, spina bifida, hemophilia, etc. And that can allow the parents to make informed decisions about the medical treatment that they're going to take. Usually this happens around 14 to 16 weeks of gestation. It is not happening. It can also happen at the 35th week of gestation, but it doesn't happen much later than that, simply because much later on there's much less fluid, and when we end up with less fluid, you have more likelihood of hitting the actual fetal tissue, which is obviously a no-no. All right, so here we have a needle. It's going to usually be guided by an ultrasound, so we have eyes on the ground inside here to figure out, well, not eyes, I guess we have sound on the ground, we have ears on the ground, so that we can see what's going on inside of here by that sonogram. And then once we send that needle in, we can go ahead and aspirate some fluid. We can take that fluid and we can analyze it again for any sort of genetic testing. Another type of testing we can do is chorionic villi testing, where we're going to take the chorionic villi and analyze it with different chromosomal abnormalities, etc. So again, it's very similar, but instead of using a needle to go through into the fluid, the needle will go in and take something from the chorionic villus. And then this is usually going to be done transvaginally. So we can either take a needle in and do it that way, or we can go in this direction from the vagina and get a sample that way using an ultrasound guided catheter. All right, so again, it can happen either via a needle or via a catheter, getting into and removing samples from the chorionic villi. Either one of these would be guided by ultrasound, again, to make sure that we do not hit the embryo or the embryonic tissue. Well, I guess we were aiming only for this particular type of embryonic tissue. All right. So let's talk about some changes that happen during pregnancy. So in the first early months of pregnancy, the corpus luteum is going to be sustained. Remember, the corpus luteum was only going to stick around for 2 weeks and before it became the corpus albuginica? Yeah, that was before it got a shot of HCG. Once it gets some HCG in its system, it's going to stick around all the way until the placenta is going to be able to take over. So that's going to allow us to maintain the uterine lining. Remember, it's going to help keep that nice thick, what we were calling an endometrium, but now we're calling it decidua. And also start inducing lactation, right? Preparing the mammary glands to secrete milk. So these are some of the hormones that are involved in that. Again, HCG is that specialty player that keeps the corpus luteum going much longer than it normally would and prevents prevents it from becoming the corpus albicans. And the corpus luteum is going to secrete progesterones and estrogens, which are going to help maintain, it says endometrium, but really it's a decidua now, maintaining that layer in the uterus during pregnancy and help prepare the body for lactation and also prepare for labor and delivery. Additionally, it's going to secrete relaxin, which is involved in flexibility of your joints, including your pubic symphysis, and helps with dilation of the cervix for the labor and delivery. We also have human chorionic somatomammatrophin, which helps create lactation, so it helps prepare the mammary glands for the lactation event. And also it's going to help enhance growth and decrease the use of glucose and increase the use of fatty acids for ATP production. Last but not least, we have corticotrophin releasing hormone, which is going to help establish birthing timing and increase the secretion of cortisol. All right, so there's a lot of different hormones that are here to do a lot of different things. Well, first and foremost, HCG has to get turned on, and it has to get turned on really early on. Why? Because HCG is what tells the corpus luteum to stay on for longer and not become the corpus albicans. And remember, if the corpus albicans happens, then we are going to menstruate and lose the pregnancy. So HCG has to be turned on pretty early on. It have a nice spike, and then it's sustained up until the point where where we have the placenta taking over. Now, this HCG is incidentally what we test for when we do pregnancy testing. We do that by way of like an ELISA, right? You pee on the stick, the urine travels past an antibody. If the HCG is in the urine, the antibody reacts, giving you a reaction on the stick. We also have a control line. So either we have 2 lines for pregnant or 1 line for not pregnant. Usually they're 2 lines parallel, but sometimes it's a nice pretty little plus to make that lady happy. It's positive, it's a plus. But really, it's just that 1 line is the control line, and 1 line, so the negative line would be the control line, and the positive line would be where the urine actually crosses. And if HCG is present, the antibody detects it. All right, but I digress. Okay, so the placenta is going to take over, right, as HCG is decreasing, and estrogens and progesterones are going to increase. All right, that's because the placenta is going to be involved in creating estrogens and progesterone. But don't forget, the corpus luteum is also creating those estrogens and progesterones. And so it's going to be creating them and synthesizing them and maintaining them as the placenta then takes over to then increase their levels, right? Let's see. HCG is going to take the place of luteinizing hormone. So it's going to continue, the LH is going to basically tell the corpus luteum to be sustained. And that's going to mean that the embryo is able to continue and remain attached inside the lining of the uterus. Otherwise, we would have a menstrual event and this embryo would just be shed. There would be nowhere for it to be embedded. All right, so I talked about all of those different hormones. This is showing you full-term pregnancy when the baby is head down and delivery is imminent. And as you can see, this tiny little thing here is a lady's urinary bladder. So she's cycling through about 500 milliliters of amniotic fluid throughout her body. She's drinking an enormous amount of water to help maintain her 1.5 times her blood supply, and her kidneys are filtering through at a faster rate than normal people, but she's got this itty bitty little bladder. So she is going to be using the bathroom much, much more frequently than other people. In fact, in the late stages, she might accidentally be releasing urine when laughing or on a bumpy car ride or when surprised because of the fact that there truly is just so much pressure and so much weight, right? This fetus and all of the fluid inside here is going to weigh about 12 to 15 pounds, not to mention any of the abdominal fat and extra weight she may have gained. So that is going to be a good bit of pressure on this teeny tiny little bladder. All right. This here, of course, is going to be our intestines. So we can see everything's displaced. Here's our small intestines. Here's our our large intestines. And then the ovaries pushed all the way in the back as well. Our fallopian tubes are going to be in the back as well. So everything is going to be, here's our ovary and our fallopian tube. They've been totally distended in where they're found right now because of the fact that the baby is pushing all of those inner organs all the way to the side. So some of those changes associated with pregnancy are going to be weight gain. Well, yeah, you've got a fetus inside there that might weigh upwards of 10 pounds by itself, and then all of the fluid. And then we also have increased protein, fat, and mineral storage in the body, as well as breast enlargement to be able to prepare for lactation, and lower back pain because we are going to have a weird, odd distribution of weight. This leads to what we call the pregnancy waddle, which is a very strange walk that pregnant women tend to do, where they go left and right and left and right, an odd canter or lumber instead of moving their legs forward and backwards. Let's see. All right, so let's talk about labor. Labor is the last stage where the fetus is actually going to be expelled from the uterus out of the vagina, and that's actually going to be the process of parturition or giving birth, right? So this is going to be dilation. They're going to be looking to see how large the cervix has opened up to, and then eventually the head is going to push through there. It's going to be expelled, and then later we're also going to have the placental stage of expulsion. So we'll have placental expulsion as that afterbirth region. All right. And labor is going to occur because of concentrations of estrogens, prostaglandin, oxytocins, etc. And again, those 3 stages are going to be the stage of dilation, the stage of expulsion, and the stage of the placental or afterbirth delivery. Okay, this image I just wanted to kind of point out to you because I wanted to show you the difference in how the fetus is respiring prior to and after birth. So circulation is going to be different prior to birth because post birth we're going to be breathing in through our lungs, so we're going to have the type of circulation that you're used to. But prior to birth, we're going to have fetal circulation, which is going to come from the placenta, which is going to result in this, it's purple instead of red, right? Our blood is actually always going to be a little bit lower oxygen than the maternal blood supply because by the time the blood gets here, the mom's going to have used some of it. So you have mixed oxygenated and deoxygenated blood circulating through the entire system because the actual site of gas exchange is occurring in the placenta itself, right? So the blood comes in as partially oxygenated and then it becomes less and less oxygenated. It's a mix of oxygenated and deoxygenated, and then it's going to end up secreting out. So basically, this fetus is surviving on lower oxygen blood. And that's actually true because, and I digress, this won't be on the exam, but the fetal blood is comprised of fetal hemoglobin. So alpha and gamma hemoglobin instead of adult hemoglobin, which is alpha and beta. And gamma hemoglobin has a higher affinity for oxygen, which it kind of has to because it has to be able to pull oxygen out of blood that has lower oxygen levels, right? This purple blood, which is not, again, fully red. So at birth, there's going to be some serious adjustments that the body has to make. So the body systems have to make adjustments, including learning to breathe, as well as the circulatory system. And so there's a lot of different things that have to occur that have to go right in order for this baby to be able to have, again, respiratory adjustments and cardiovascular adjustments have to occur in order for this baby to be able to breathe on its own. All right. So now we're going to talk about lactation. Lactation is another response that is going to rely on a controlled condition. So basically, the physiological response to the suckling of the baby on the nipple will cause the touch sensors to send nervous impulses into the hypothalamus and posterior pituitary, which causes the release or the contraction of the myoepithelial cells, which causes the release of the milk or milk ejection. This is called letdown. Milk availability is going to encourage continued suckling, so touch centers are going to release more, etc. So it's going to be a feed-forward mechanism until the baby either ceases to suckle or the milk has been entirely ejected, okay? So this is, again, a positive feedback loop. It's actually kind of unusual. I know we normally see negative feedback loops. So these are the hormones involved in lactation, including prolactin, which is going to come from the anterior pituitary gland, and also oxytocin. Oxytocin is going to be the love hormone. It allows you to feel at ease with your loved ones and your fetus and your baby during breastfeeding. It allows you to form a sense of attachment. It also is what's going to make you feel intense or worried when the baby cries, because it's going to basically attach you to the baby. Oh, all right. Here's inheritance, all normal. All right, I apologize. All right, I'm going to stop here then because this is all of the stuff for inheritance that I wanted to put up earlier, but I guess wasn't present. So inheritance got duplicated. All right, so I'm going to go ahead and stop here then and go ahead and ask if anybody has any questions. Do you guys have any questions? Nope. Actually, there's just one of you. So, all right. I appreciate you sticking around to the end there. I'm sorry other people had to go. You have a question?
Yes, I do.
Sure, shoot.
Are you actually recording at the moment?
I am. Would you like me to stop?
Yes, please.
Sure thing. Okay. Thank you very much for attending our review session, and I will see you guys shortly when we get to our lab review.